let's look at how drugs affect this process, okay? So I indicated that one method by which you can depolarize a neuron is if we look at a particular type of sensory neuron, like the pain neuron, neurons that are going to be carrying information about pain. And what they're really carrying is information about destruction of tissue, right? Of something that has burst, the cell membranes of cells, of tissue, and promote or provoke the release of proteins that are normally inside cells so that they are now present out in the extracellular space, right? And these cells, will have specific protein receptors that are embedded in their membranes that'll bind these proteins that belong inside cells, right? And then they will allow for sodium to flow, and they'll open sodium channels that'll depolarize those cells to -55, and then all the voltage-gated sodium channels will open, and then you'll start getting a current, going down from one end to the other, and you'll feel pain. So think about yourself at the dentist, right? And somebody's there with some kind of a little dental kind of probe, and they're poking away at your tissue, and you're bleeding. There's been some tissue damage, right? There's some cells whose membranes have burst. So what the dentist will often do is they'll apply a local anesthetic, right? Something like, let's say, lidocaine, okay? Lidocaine, it turns out, is a chemical. It's a drug, right? And what it will do is it'll actually, has a certain structure, a certain shape. It'll actually come over to the voltage-gated sodium channels on your pain neurons, and actually on all the neurons that are present in this area, and it'll attach to them. It'll interact with them. It'll bind with them. And remember, when one substance binds with something else, it changes its shape. So the binding of this, lidocaine with your voltage-gated sodium channel prevents it, from responding as it normally does when you get to the threshold potential of -55. So at -55, it will not open. It'll block it. So even though the dentist is, tearing away at your gums, and the information, should be sent, the information is cut off at the source, and you feel numb and pretty happy, actually. There are other drugs that can affect, propagation of the action potential. There is saxitoxin, which is a chemical that's produced by red tide, which is this kind of algae bloom that moves up the East Coast in the summertime. And you're not supposed. You have to avoid shellfish because they will end up, being infected with this red tide, and you can be exposed to saxitoxin. And saxitoxin also will block voltage-gated sodium channels and prevent neuronal transmission. This can have significant effects, for example, on heart function, or control over muscles if you're not able to send electrical signals along motor neurons. Tetrodotoxin is actually from the puffer fish, actually, which is a delicacy in Japan, but it has to be cooked to the right temperature and for the right length of time. And apparently, sushi chefs who prepare blowfish actually need to go through quite significant training, because what they try to do is they try to leave enough tetrodotoxin in the fish to give you a slight numbness in the mouth as you eat it, but not enough to actually stop your heart from beating. So you really want a good chef if you're going to eat tetrodotoxin. There are other poisons or drugs that can influence the process of action potential transmission. It turns out that buttercups, never eat a buttercup. It contains aconitine. And then lilies also contain something called veratridine. And I think aconitine actually makes sodium channels open too quickly. So they open at a lower threshold. So instead of -55 as the threshold potential, you might have sodium channels that open at -62. So they're much more responsive, and you're far too responsive in terms of generating action potentials in response to stimuli. And I believe the lilies actually, that particular compound from lilies will actually bind to the voltage-gated sodium channel and cause it to stay open for far longer than 1 millisecond, which will interfere with the appropriate transmission of neuronal signals as well. So drugs can influence, transmission of the action potential, have significant effects by acting, at these specific receptor sites. In this case, we're talking, primarily about the voltage-gated sodium channel.